Okay, so first of all, our screen is broken. So I found that out from other people that were in here before us. So hopefully that will get fixed today. Still getting some feedback. Lots of technical things to get worked out today. Hopefully, as we go forward, it won't be such a problem. But I am trying to record on Zoom, so that's why I'm getting a little bit of feedback. That's going to be really annoying. All right, well, we'll go forward. Hopefully that won't be too annoying. The second thing that's annoying to me is I love seeing all your faces. I was exposed over the weekend, and so I'm going to max up for a little bit. Hopefully I will still be here all week. First time my household has had COVID, which is pretty remarkable, but that's what we're dealing with at my house this week. So I am Professor Leonard, and hopefully you are here for microbiology, because that's what I'm going to talk about today. Welcome. And again, that is not what I wanted to have projected. Oh, man. Oh, there we go. Let's try this again. All kinds of fun things today. Oh. I think that's at least. I can't get it to recognize this. But... All right, well, sorry about this. I will have to come in another time to figure this out, which is almost impossible to do. Trouble is, my cursor doesn't go on the room thing. All right, so I think what I'll have to do, I posted the PowerPoints, and I'm going to talk through the PowerPoints, and this is going to be really difficult without you actually seeing the PowerPoints, but we're going to kind of piece together and then do what we can. And obviously, I'm not very technological. I have taught this class quite a bit and encountered many different rooms, but I cannot get that to go to the right place, unfortunately. It will be saved on the Zoom meeting. So I am trying to record lectures. That's why I'm having problems, because I am trying to do a little bit of everything here. A little bit about me. What I'm planning to do is just kind of go through the syllabus just briefly. I need you to all read the syllabus in its entirety. It's going to hopefully answer a lot of questions that I won't get to today. But I'm just going to hit the high points there. And then we're going to start right in with a little bit of contents. Okay. My office hours are on the syllabus there. With my office hours, I'm trying to work around. I also teach 3 labs. So of course, those micro labs meet twice a week. One of them is directly after class today. So unfortunately, I can't really stick around after class very easily. So I do have a lot of gaps in my schedule though. So don't be afraid to come see me a time out in my office hours, especially if it's for something kind of brief. Okay, so I'll be there for sure during my office hours. If you want to set something up another time, we can definitely do that. We can do it by Zoom. My phone number is now a Zoom phone, and that's new for all of us faculty. I don't know quite how that works, but you're welcome to Zoom me during my office hours, especially as well. I was going to show you a picture of my family. Of course, that's not going to work. I just briefly about me. Like I said, I've taught this class this is my 9th year. So I probably taught it like 12 or 13 times, something like this. We did change textbooks this semester. So it's kind of like the first time all over again. I had to retool quite a bit. They didn't tell me till the end of July that I was going to be teaching this class this semester. I thought I had a little more time to kind of work all the kinks out. So bear with me. The main reason that we changed is to benefit you. So you didn't have to pay for a textbook. That, of course, we're going with the OpenStax, and I see about a third of you as of this morning had registered for that. About a third of you are in that already, so that's good. Somebody unplugged it. I don't understand where I'm getting that feedback. The big drawback in my mind with changing textbooks is the old textbook had a huge repository of study helps and animations and a lot of homework. And so it was easy for us to kind of pick and choose what homework we wanted to give you. The homework now that is associated with the OpenStax textbook is, it'll be at the end of the reading, and there's not a lot of it. I mean, maybe you see that as a good thing, maybe not, but they're kind of very straightforward questions that you should be able to find in the reading, or hopefully like 90% of the time from what I've talked about in class. So we'll get to that a little bit more. In terms of introducing myself though, I have 3 kids that are adults. My youngest is out of college now. My oldest is in the Navy, in med school in the Navy. So I've been through that whole med school process with her, and I hope I can be a help to any of you that are hoping to do that. Our middle child lives at home with us. She has autism. So that's my kids. I live close by, and my husband teaches in chemistry. He is Dr. Leonard. People sometimes call me Dr. Leonard. Also, I'm not a doctor though. So Professor Leonard is probably the preferred thing to call me. I just have a master's, so. All right, the course code is there in the PowerPoint. It's also on a tab on Blackboard. You'll need that code to go into Top Hat to access this course. That OpenStax textbook, we have taken that and changed it a little bit. We've chopped some things out. And so you'll want my specific code. I've written some homework questions just for you all that would be different from somebody else taking another section. So look that up here in the next couple of days, and just so you don't have any trouble. I have run into some hitches in setting up the course, and when I contact the chat for Top Hat, they've been very helpful right away in getting things working again. So by all means, I may be able to help you if you have technological problems. Might be better off just going to Top Hat and saying, you know, what your issue is. Jumping right into how the class will be graded. If you've taken a look at that at all, it's mostly exams. I'm kind of old school that way. Most of the other people that teach this class, your score is mostly exams. We do have a small homework component that's worth about 10% of your grade. In the first exam, I have weighted a little bit less. I have found that oftentimes students bomb the first exam. They just weren't prepared for knowing things as well as they needed to know. So I've made the first exam worth a little bit less. we're going to have that. Hopefully, I'm going to beat all your other classes, and your first exam will be in micro. So I found that you usually get that first week of this, that 1 week of the semester where all your classes have exams at the same time. I'm going to try to do it a week ahead of time and beat your other classes. So for instance, the lab exam, which a lot of you are in micro lab, that will be the following week. So our first exam weighted a little bit less material than the other exams. Hopefully to help you. The final is weighted a little bit more, and I know that puts more pressure on you to do well the final, but I also want to reward those of you that are kind of doing better and better as you go along. You know, if you ace the final, then my gosh, I hope you get a good grade in the class. So the homework, the way that I'm planning on doing it is there's really not a ton of homework points. I actually haven't counted up how many questions I have in there. I'm planning on weighting that if you get 80% of the homework questions right, then you get all of the homework questions. So right now I have the homework as 60 points. If you get 80% of those questions right, you get all the 60 of the homework points. And I also have padded that. I've thrown in some participation points just for doing the homework. And like I said, I don't think that's going to be a huge time sink for you. But I didn't want people to spend so much time on the homework that they're trying to get every little point right. It's fine to guess every once in a while. The reason that I want you to do homework is just to get more practice. Like, you need to see things several times before you really kind of know it, before you really get it in your long-term memory. So, lectures, homework, reading, see things several different ways, several different times, and hopefully that will be helpful to learn it. I know that some of you have probably heard things about this class. Maybe some of you are kind of nervous about this class. I do have, just some recommendations. I am posting PowerPoints. So those PowerPoints, and again, this lecture is going to be different than all my other ones, hopefully. Those are just outlines. So you'll need to take notes during class. Some students have complained you have to take too many notes. Other students have complained you don't take enough notes. I try to hit the center there. Everybody's a little bit different, but I'll be writing things in on my tablet. So if you don't have a tablet, that's all right. You can type things in on your laptop. Students always used to print those PowerPoints off and write on them. There's a number of different ways that can work. But you will need to take some notes. I will be writing in things that are important. If I write something down, then you're going to want to write it down somewhere. I do have learning objectives posted as well. Okay, so if you've had a chance to access that, it's kind of a long document. It's like 17 pages, I think. So what basically I've made learning objectives for every chapter, and I have retooled this a little bit to match the kind of the new textbook and the new order that we're doing things. This looks a little overwhelming. You may have heard the phrase inch by inch, life's a cinch, yard by yard, life is hard. All right, so take these, look at them as we go. Okay, don't wait till the night before the exam and say, oh my gosh, I got 4 pages of objectives. I can't do this. Yeah, that's really hard. So kind of keep up with these. Some students find it helpful to kind of go through and answer all these. You may not need or want to do that, but I will say that I go through these, and this is what I look at when I make an exam. I'll say, okay, I need a question that falls under this objective. Okay, yep, I got one there. So then I make sure that I kind of, spread the exam around a little bit and covered everything and covered things that we talk about. Test you over things that we talk about. So there may be some stuff in the textbook that we don't get to. You don't need to worry about that. Hopefully the homework questions reflect things we talk about in class as well. Okay. So I would encourage you to do those. Like I said, there's not very many for each, maybe for each chapter, there's maybe like 10 or 15 homework questions. It's not a lot. So I would encourage you to do that after I've lectured over it. See if you can answer them without even looking in the text, and then it's there for reference if you need it. Terminology. With any biology class you take, there's going to be a lot of new terms to learn, and I have tried to boldface those terms that I think will be new in the learning objectives, just to kind of emphasize that. And if I'm using a term that you don't know what it is, either stop and ask me about it, or look that up, because you don't want to just keep glossing over it, because we're gonna build on that terminology. So I encourage you to pay attention to that. And sometimes students ask me, do I really need to read all the homework assignments? No, people learn different. There's going to be certain sections where I'm going to really encourage you. I really suggest that you read this, because this is complicated and you probably want to see it a couple different ways. But okay, so I would use it more as a reference. Like if something I say that doesn't make any sense to you, go back and look there, and then ask me about it if it's still not making sense. So. All right, so what are we going to be studying this semester? In the syllabus there, I have mapped out about the pace that I think we'll be going. The last few years I have taught this class Tuesday, Thursday, so I've lost a little bit of my pace, what I think we'll be doing. But the exam dates I'm planning on keeping pretty firm. We might get ahead or behind, but I'll just adjust the content that's covered on those exams. So I try to keep those pretty firm. We start out just all the basic things that you need to know about microbes. Microbes meaning things that are small, too small to be seen with the naked eye. So we're going to talk a little bit today, a little history, a little taxonomy. I'm not sure how far we'll get into that. And then we're going to kind of go out of order and talk a little bit next time about culturing microbes in the lab, because we do try to line up a little bit with what you do in lab. And then just kind of basic stuff like what are microbes, what their cell structures look like. We're going to talk about different kinds of microbes, how they grow, how they get kind of things they eat. And then we get a little bit more abstract, like what's their metabolism like and how do they reproduce, what are their genetics. And that gets us through about halfway through the semester. Of course, we're going to talk about viruses. Sometimes I talk about different points, but that seemed like a good time to talk about viruses. And then we get into the really clinical bent. So this course is really more for those that are interested in going in the medical field. We have a real clinical look to things rather than environmental. Okay, so there is an environmental microbiology class over in biology. They focus a little bit more about, say, the microbes that are in the soil or in the water. We are most interested in ones that cause disease. So the second half of the course, we're thinking about how to kill microbes when they might be harmful to us. We're thinking about what are antibiotics, how do we kill microbes in our body, and then kind of how do microbes cause infection, what are the tools that they use, as well as our body, how our body systems work to kill microbes. So we're going to spend a good bit of time on some immunology. And then basically at the end of the semester after that, we pick out some particular pathogens, some disease-causing microbes, and talk about them a little bit more in depth, like the microbes that cause tuberculosis or staphylococcus or streptococcus, some of those skin infections they cause. I kind of pick different ones, different semesters. There's some things particularly in the news, and I try to talk about that, like measles or Ebola. Maybe we'll talk about monkeypox this year. Kind of tired of talking about coronavirus, but we'll see if there's anything new to discuss with that as well. So most students like that kind of end of semester stuff because we kind of review some of the stuff that we've done all semester about those specific pathogens. So, all right, any questions, kind of introductory stuff? I have put in the syllabus some little, hints on studying, ways to study actively. If you're a procrastinator, there's, some things that you can do to try to motivate yourself. But make sure you look over that syllabus. All right. I wonder if I used the wrong chord. I don't know. I don't know what I did. Okay, so we're gonna get into a little content of chapter 1 here. Again, I'm sorry, I'm probably bore you to tears when you don't have anything to look at up here, but hopefully you can bring those up on a device. We're going to talk a little bit about some key contributors, historical figures in microbiology, a little bit of taxonomy, which we don't spend too much time on that, but as well as types of microorganisms. So, and again, I guess hopefully this is recording the way I have meant it to. You can always look back at the recording if something I say is really unclear. But first glimpse of microbes. Anybody know the guy that we give credit to for developing the microscope? It's kind of a long, cumbersome name. Okay, Antony van Leeuwenhoek. And again, I'm gonna just write that down. A lot of people in West Michigan have Dutch roots. So van Leeuwenhoek, he was a Dutch cloth merchant back in the 1600s. Yeah, I could do that. Thanks. I think that I will just turn that projector off. So. Antony van Leeuwenhoek, and he was mid 1600s, just to kind of 1650s, just to give you an idea of when he was alive. Dutch cloth merchants that is given credit for really the first workable microscope, and that seems like, why would anybody make a microscope that was a merchant? Well, if you think about stereotypes of Dutch people, they're clean, tall, maybe blonde. Also, let's say thrifty. Okay, you might know Dutch people that were very thrifty. And he wanted to make sure that nobody was taking advantage of him. If he was buying cloth that was supposed to be high quality, he wanted to make sure that it was high quality. So we got really good at making these lenses so that you could use the cloth. And the first microscope that he made is really a really kind of bizarre looking thing. It kind of looks something like this. It had a little lens right here, a little post that you could, put a fly on or whatever he was trying to look at. It doesn't look anything like the microscope we have today. We would call this a simple microscope because it only had one lens. And he looked at all sorts of things, pond water, bodily fluids, and he wrote up what he found to the Royal Society of London and was recognized as a scientist of his day. Unfortunately, he didn't share with other people how he made those lenses. He kept it a secret, and so we didn't really suddenly take off and make all these great microscopes for many, many years. Another, along those lines, Robert Hooke, he was a contemporary of Leonhood, and he is given credit for first coining that term cells. Okay, he looked at some dead pork and it's on little compartments, and he called those little compartments cells. We didn't really understand that that was kind of the basic unit of lice, but we're kind of giving him credit for also having some workable microscopes. His microscopes actually had several lenses in them. So we call that a compound microscope when it has more than one lens. And again, there's some pictures in your book there. It must have been fabulously difficult to get a good source of light for those microscopes, using a flame, maybe some mirrors. It must have been really tricky, but they actually got pretty good magnification. Leeuwenhoek was able to get magnification equal to like medium power on our scopes upstairs. So pretty decent magnification, more than what you would think. Okay, so now we're going to jump ahead maybe 100 years to what's considered the golden age of microbiology. So if we say from about 1850 to 1910, we consider that's kind of an era where lots of advances were happening really rapidly in microbiology. So two people that we give a lot of credit to, Louis Pasteur and Robert Koch. They were contemporaries during this time, 1850s, 60s, 70s, doing a lot of their work. Louis Pasteur was a French scientist. Robert Koch was German. They were on opposite sides of that Crimean War, hated each other, so they worked on very similar things, but they didn't collaborate. Unfortunately. Louis Pasteur is most remembered for a number of things. He really helped us understand the process of fermentation, and he came up with a method that we call pasteurization. Oops, right, kind of small there, to save that winemaking process in France. So in France, they were having problems with our wine getting contaminated, and that was changing the flavor. It was very bitter. And he figured out that it was the yeast that you wanted to ferment the grape juice, not the bacteria. The bacteria were giving very acidic end products and spoiling the wine. So he came up with a process to heat the grape juice and get rid of the bacteria, called pasteurization, and then add the yeast that was going to give the alcohol that you want. He also developed vaccines for anthrax as well as rabies. So some of our very early vaccines. Where Robert Koch was much more concerned with what causes disease. So we call that etiology of disease. Okay, what causes disease? He figured out what caused anthrax, that it was a bacterium. You know, up until that time, people had all sorts of crazy ideas about what causes disease, like foul air and all sorts of things. Tuberculosis, he figured out the causative agent of tuberculosis. Up till that time, it was thought that tuberculosis was a cancer. So a lot of important things there. Moving on, then John Snow. He's considered the father of epidemiology. So that's kind of a hot field right now. Epidemiology, the study of kind of where, when, why, spreading of diseases. Epidemiology is really a lot of asking the right questions, doing a lot of groundwork, a lot of statistical analysis. There was a cholera epidemic at that time. Again, don't worry about years, but in the 1850s, that he went door to door and asked people, is there someone sick with cholera in your house? And he made detailed analysis of that and realized that the people that had cholera were clustered around one of the water pumps, the Broad Street pump. So he suspected, obviously, that there was something in the water that was making people sick. He took the handle off that pump and the epidemic subsided. So Again, he's thought of the father of epidemiology. And then in terms of preventing disease and infection, a couple names: Ignaz Semmelweis. He was a Hungarian physician that in 1848 noticed that many mothers that gave birth at the hospital, maybe around 10 to 15% of them, actually developed this infection called childbed fever. And that was essentially oftentimes deadly for the mothers. So this was a huge problem. And mothers that gave birth at home, a very low percentage of them, maybe only 1% got this infection. So he started advocating hand washing. Just simple hand washing before physicians would help mothers deliver their babies, and that cut the rate way down of infection. Unfortunately, he didn't really understand why that helped. Doctors at that time were, doing autopsies on cadavers in one room and going to the next room delivering babies. So Semmelweis said, well, there's something in those cadavers that's making the mothers ill. They didn't really understand what it was. And because they didn't really understand what was causing those mothers to become ill, this wasn't well accepted, even though he documented it. But people took note of it, and John Lister, about 30 years or so later, I think this was like 1880s, he took note of that. It must have, someone must have written it up somewhere, and he had advocated hand washing with carbolic acid, which is phenol. Okay, now phenol used to be kind of the gold standard for disinfectants. We don't use this hardly anymore. You don't know why? Really caustic. This really cut down on infection during surgeries, but for the surgeons that were washing their hands in phenol all the time, it made their fingernails fall out. They lost their hair. They sprayed this in the operating rooms, and the surgeons had respiratory problems. So, it was a good first step to understand, some hygiene things that we could do. But, thankfully, we've come up with much safer things to use now. Right. Also, in terms of preventing, so we have good hygiene. That's going to prevent infections. Another big step in preventing disease was vaccines. Okay, and Edward Jenner is given credit with developing the first vaccine. Who knows what that first vaccine cured? Smallpox, yes. And in the news a little bit lately because monkeypox is related to smallpox. Smallpox carried about a 30% mortality rate, so huge scourge for hundreds of years. He noticed, or at least he's given credit with noticing that milkmaids that got cowpox, very a milder infection than smallpox, very rarely came down with smallpox. So he thought of taking the scabs from the lesions from a milkmaid with cowpox, and he actually cut them into the skin of a 10-year-old boy. Not exactly how we do clinical trials today. Not exactly. And then a couple weeks later, he actually took the scabs from somebody with smallpox and injected them into that same boy, or cut them into the skin. He didn't get smallpox, so it became this vaccination, vaca, cow, from a cow, sort of, became a very common practice. Vaccinations are way safer now than they were then. They had the much, higher chance of getting some secondary infection back then. And another name that is probably not as familiar, Paul Ehrlich. When we're thinking about trying to treat infections, ideally we're going to prevent them by having good hygiene or vaccines. But we want to treat them. Paul Ehrlich, he's kind of thought of as the father of chemotherapy. And when we say chemotherapy, we think of treating cancer, but really that term just means, any drug used to try to cure some disease. He was searching for the magic bullet. Okay, on the battlefield, if you had a magic bullet, it would kill the enemy and not, harm your friends if they got hit. So he was looking for a drug that would harm the bacteria or the virus or whatever. By this time, this was, maybe around 1905. We understood that what kind of things caused infectious disease. And he was looking for a drug that would harm that pathogen and not harm the host. So a magic bullet. And he experimented with a lot of different arsenic compounds to try to find one that would cure syphilis. And he found one, and again, of course, arsenic is pretty toxic to our bodies. So it was not a great thing, but it was a start. And for many years, people were treated with syphilis by that compound. We call it salvarsan. Eventually, we realized that you could cure syphilis if the patient got a really high fever. So before we came out with antibiotics, we were actually treating syphilis patients by giving them malaria. You get a really high fever with malaria, and it would cure the syphilis. Hopefully, you pick a kind of malaria that's not a bad one. But, okay, so those are kind of that's my little kind of history lesson. There may be some other names in the textbook, but those are the ones that I particularly wanted to emphasize. And the next section gets into taxonomy. So I got another name of somebody that was kind of an important figure in helping us to kind of organize or what we call the father of taxonomy. And his name is Carolus Linnaeus. Okay, he was a Swedish botanist, okay, Carolus Linnaeus. And we're going back in time again to close to Leonhood's time. So he lived from about 1700 to 1780 something like that. And like I said, he's considered the father of taxonomy. Okay, and what do we mean by taxonomy? Okay, taxonomy is considered a science of classification. What's the other thing? Identification. As well as nomenclature. And those things, they all kind of sound like about the same thing. But what I mean by that, classification is when we put organisms into groups. Okay, so we classify them with other organisms that are related in some way. When we ID an organism, that means we decide what group they belong in. So that's deciding what group the organism belongs to. And nomenclature refers to naming rules. Okay, so sort of in order for scientists to be on the same page, they kind of have to have this agreed upon set of ways that we name new organisms as we discover them. Okay. So Linnaeus, he's the one that suggested that we kind of have this hierarchical system of all of the organisms. All right, so you may have learned this in high school biology, how we have organisms in terms of kingdoms and phylums, class, order, family. That's how we organize them, and then genus and species. Okay, so I learned the mnemonic. King Philip came over for grape soda. It helps me remember what order those go in. Okay, so at that time, he said, okay, all of life belongs in two different kingdoms, either plants or animals. Okay, and obviously, we've been revising this system ever since. Right, we have a lot more kingdoms than what we used to. Kind of go through the revisions on that. Linnaeus said organisms are considered to be in the same species if they interbreed. And of course, that doesn't work so well when we're talking about microorganisms. Today, we say that if two organisms have 70% homology in their DNA, then they're considered in the same species. But again, Linnaeus said that they interbreed, they're in the same species. Okay. Okay. I better check and make sure that's mostly saying what I meant to say. Oh, and how did he put those in those groups? He based them on shared characteristics. Okay, so if animals both have fur, maybe they're going to be in the same phylum, for example, or lay eggs, or different species that he could recognize. He also came up with lots of different rules for naming. So that's, remember, nomenclature. He came up with what's called binomial nomenclature. Okay, so that means naming rules or nomenclature where they have actually two names binomial. All right, so he said, hey, every organism is going to have a name that has two names, the genus and species. And that genus and species is going to be kind of written in the language of Latin. Okay, so you don't want a Spanish name and a German name and a Japanese name, something all different. We all agree on that genus and species, and that's the language of science, Latin. So it's going to have a Latin sounding name. The genus is supposed to be a noun, and the species is supposed to be describing that noun or an adjective. That's a little bit looser with that. For example, the organism Escherichia coli. Okay, that's named for Theodore Escherich. He's the one that discovered it. Coli means of the colon. Okay. Grammar is something that I emphasize, and now that you're micro students, you should get in the habit of writing your genus and species correctly. Your genus should be capitalized, and your species lowercase. Okay, so like E. coli, you're going to capitalize that, and the species is going to be lowercase. If you're handwriting it, you got to underline it, right? You're going to type it, when it's appropriate to italicize. Okay, so underline. Okay, my lab students, I get after them and take points out, but they don't write those organisms' names grammatically correct. Okay, when can you abbreviate it if you've already written out the full genus name, like Escherichia, earlier in your paper, then you can start to abbreviate it after that. Okay, E. coli is so common that we often don't bother with writing that whole thing out. All right, so those are naming rules that we'll want to kind of get used to and keep practicing. Like I said, taxonomy is continually being updated as we learn new information about organisms. About mid-1800s, let's see, write this over here. Really the next major revision, a guy named get this right, Haeckel, H-A-C-K-E-L, Ernst Haeckel. He said, well, we need to have more than just plant and animals. Let's add the protists. Okay, they were, again, this is kind of mid 1800s. Their microscopes are getting better. They could see there are all these protozoa moving around. So he said, let's add the protist kingdom. And then later on, he added the Monera kingdom, which basically is what we would call prokaryotic organisms. Okay, so protists are eukaryotic, Monera are kind of an old-fashioned term for prokaryotic organisms or just bacteria even. So he made a new tree of life that had the plants, the animals, and the protists. I'm not sure where he put those Monera in relation to that. And then really there's a nice little kind of sum up diagram in the textbook there that has one other revision, and that's in 1969. They kind of give the nod to Robert Whittaker. He added another kingdom, the fungi. So up until that time, they considered plants and fungi the same thing. What's the main difference between plants and fungi? There may be some differences there. There's another big thing, yeah.
Plants need light.
They need light because they do what? Yes. Plants photosynthesize, fungi do not, right? So that's kind of a big difference there. We'll talk more about fungi later. But those were the kingdoms that I learned when I was in my high school biology class. Whittaker's kingdom, he had 6 kingdoms or whatever, 5. But I think that's a good place to stop because I'm tired of doing it this way. We have another revision that we'll get to next time. We have any questions before we go? Hopefully I'll get my stuff. All right. Have a good day. Thank you.
Yeah, it's kind of nice. There's a lot of it's really great. It's called this place called the Shoreline Center in Grand Haven. So I travel in the center and I also travel to families.
Oh, yes. I'm sorry, it's so fast. Okay, there's different variations of that. Yeah, I'll have to go in and probably bring all that still. I'm sure. Yeah, that will all be in one point, yeah. So this was a total fail. But it should get better. Yeah.
I'm also a pre-dental student, so looking forward to that.
Yeah. For sure. Okay, good. Hi.
I'm supposed to tell you hi. Do you know Maggie Myers?
Oh, of course, Maddie. Yes.
So I'm her fiance, so recently.
Oh, okay. Congratulations.
Just wanted to say hi.
Yeah. What is Maddie doing now?
She is just working right now at Spectral. She's applying to PA school next.
Oh,
summer.
she decided to go PA.
Yeah.
Okay. Okay. Well, good. What was her name again?
Jacob.
Jacob. Okay.
I'll see you in a little bit.
Oh, okay. Good.